Thank you, Thomas. I think you need to stop sharing so that I can. Okay, perfect. So can you see my slides now?
Yes.
Perfect. So, sorry. I can't see anything. Okay.
Lalith, it was cool. We saw the slide.
Yeah, cool. So thank you, Thomas, for the nice introduction regarding the total body PET. So as Thomas told you in the previous...
Now it's a blank screen, Lalith.
Again, a black screen?
Now it's a blank screen. Before it was better.
Okay, let me share it again. Yeah, is it?
Yeah, we see it. Yep.
Okay. So as Thomas told you, total body PET is something very new to us in the sense that it can cover the entire field of view. As you have listened from the previous presentations, like everyone is interested in looking at the interactions between different tissues. But now we have actually an opportunity to see how different organs interact with each other, because we know that organ systems actually talk between different organs and they kind of maintain this homeostasis. The concept is the following. Once we put a subject inside a total body PET, we would like to do something called the spatial and temporal network analysis. This is not something new. I'll tell you why this is not new in the following slides. But the goal is we would like to see how these different organs talk with each other in a normal patient. And we would like to build something called a normative network, which we are showing here. A normative network is nothing but a network which is comprised of various healthy volunteers. And if you take a particular network, it will actually tell you how a normal person's organ should communicate. And this normative network is quite powerful because when a patient is being scanned, he or she will have his own patient network where you have certain kind of abnormalities. Hypothetically, they should have some kind of abnormalities between the organ interaction. And our final goal would be to compare this normative network from the healthy volunteers with a patient cohort and see what organs are miscommunicating. Because right now there are so many diseases which have been proven to have inter-organ communication. For example, you take cancer, cachexia, and also the more recent COVID. All of these diseases have shown to play on this region of multi-organ interaction. So being an engineer, I tend to build roadmaps, and this is something which we built for our particular project in the sense on how to probe multi-organ interaction. So for the first phase, it sounds simple, but it's called multi-organ segmentation, because once a patient is scanned, you will have the entire field of view, meaning that you can see all the organs. But if you want to quantify the signals in these organs, you need to define a volume of interest. So recently I found out that for making these volume of interest for all these organs that we are interested in, it'll take around 9 hours for an experienced radiologist. And as you can tell, this is not the most exciting job because it's not worth their time. So we planned on doing an automated multi-organ segmentation, being inspired from this autonomous vehicles where they can actually segment the road into multiple scenes. The second phase is once we have these signals from all these different organs, how do we make sense of it? And we have this principle in our lab that we don't like to reinvent stuff. We try to reformulate. And we looked at modalities which had similar issues. For example, EEG, they measure different signals from different regions, and they do it simultaneously. So they have developed a plethora of techniques and they are much more mature than us. Similar, likewise, ECG. And if you look at this, the imaging domain, fMRI has been thriving from 1995. They have been producing so much techniques where you can make use of these simultaneous synchronous measurements and make sense out of it. So for phase 1 and phase 2, we are kind of equipped on how to deal with this. And we spend most of our time in doing that. Phase 3 is kind of the part where we actually struggle because this is so new to us. We are not used to network analysis before, but the concept, as I explained you, we like to visualize these abnormal interactions between the networks. And this is where we are. We are currently working on phase 3. So phase 1, phase 2, and phase 3, all these three combine to form this enhanced framework, which is a web framework which we are currently building. And once we have this particular framework, the basic goal of this framework is to generate or validate or disprove hypothesis. So that's why the phase 4 is going to be like the engineers or the physicists discussing with the doctor and see if the data actually makes sense. Basically, the entire framework is kind of an exploratory tool, but pushing through things in a very fast way. And for phase 1, we actually used an artificial intelligence approach where we segmented different organs from the low-dose CT. And this is a thing which we do in our lab, that we name all our tools in name of animals. So this one is called MOOSE. The abbreviation is multi-organs objective segmentation. And we always need some kind of visualization tool. And we have to thank kitwest and BTK for making our job much easier, because all these training segmentations that we created were done with ITK Snap and also with Slicer. This made the delineation job much easier. And once this is done, we always validate, and also the training is done, we use our AI algorithms prediction to visualize the results. And this is also being done with Slicer and ITK Snap. So this is one of the tools which we often use. The second part is the exploratory analysis. Once we have the segmentations, as molecular imaging people, we don't really care about the segmentation. We want to look at the quantitative value for each and every organ. So as you see in the previous slide, there is nothing very innovative when it comes to visualizing the results. But I still remember the first time I showed these plots, the reaction of my lab members were incredibly funny, because they have not seen this before. Seen these kinds of plots before. So for us, this plot was the best to see every aspect of the organ, in the sense that violin plots are very beautiful because they highlight the distribution. They also tell you the standard deviation, mean, and all the factors together. Previously, we used box plots, but I think the distribution played a major role here. So in this particular plot, we just wanted to see the difference between male and female healthy healthy cohort and their organ values. So if you look at these, the x-axis, these are the different organs, and the blue represents the distribution of female cohort who are healthy, and the gray one corresponds to the distribution of the male cohort. And the interesting thing is you can already see that the brain values for female is much higher than male, meaning that they have a very active brain uptake of glucose. So this already tells you a lot of details when it comes to the underlying physiology. And we extensively use Plotly because it makes the whole thing easier, and we can pipeline it. And it also has an interactive backend, which we really benefited. And I just wanted to bring out this plot because I have made this is a plot between two different cohorts. The first, the pink one is actually the baseline, which means that the patient before getting any treatments, and the gray one is actually the patient after getting the treatment. So we wanted to see how breast cancer patients' physiology changes before and after treatment. So the before is the one in pink, and the after is the one in gray. So if you look at these change in values, I have highlighted in orange, you can see that there is a clear decline in organ function or the organ uptake. But the violin plots tell much more. For example, there is a change in distribution in spleen and thyroid. For example, in spleen, you have like somewhat a distribution which seems monomial, where in the sense after the treatment, it kind of spreads out more. And the same is on thyroid as well. So for us, these kind of distribution plots tell us a lot of stories on how to link physiology with the organ uptake values. And the final one, which I want to emphasize, is that the TotalBody PET not only takes a particular snapshot, it can also take a movie. So for example, I can take multiple snapshots of your entire body, and I'm going to show you a particular data analysis that we did. And for you guys, D3JS might be something very common, but this was really new for us, and this was a total game changer. So what we did is we actually took all these time activity. So these are the time activity curves, which basically means I put a particular volume of interest on these movies and calculate the values as a function of time. fMRI people do this all the time, but for TotalBody PET, this is new. For us, this interactivity, again, is very new in the PET community because we merely look at blobs all the time. We don't really go for dynamic visualizations or something which is interactive. So for example, if you look at the heart, which is the yellow one, and if you look at the aorta, which is the blue one here, you could see that the heart uptake occurs much earlier than the aorta. This already highlights the physiological circulation that happens because blood comes first to the heart and then it pumps up through the aorta to the other organs. So these kind of informations, we can visualize it clearly via these interactive plots. But what we were much interested in was with establishing a connectivity between different organs. Like, how are they correlated? How are two organs correlated? Are they bidirectional? Are they unidirectional? And things like that. So the simplest way would be to make a heat map based on their temporal correlations. But then we look at it, it's not really visually appealing in the sense that our final customers are the doctors and they don't really like seeing such visualizations. So we adopted something called this chord diagram. We used D3 as a backend, and we tried to make these chord diagrams and to see this connectivity. And for example, again, if I take the example of heart and lung, you see there is a connectivity between these two organs. The thickness of these Bezier curves actually tell you the strength of the correlation. And this link can be clearly explained via the systemic circulation. So for us, we wanted to see what kind of organs are connected with each other temporally on a very coarse scale. And this proved to be a very cool technique. This might again sound quite basic for you, but this is for us like a Prometheus moment, like where we saw fire for the first time. I'll get back to my slide again. So finally, I just want to tell you the things that are happening in our field. Our field has actually moved a lot from the 1950s in the sense that you saw the images at first. They were like they were really bad. And then there were so many advancements in this detector technology to make the devices much better. And then we also made like great strides in reconstructions where the images are very beautiful. And the final cherry on the top was increasing the field of view, because now you can measure simultaneous or like multiple organs simultaneously. And till date, this is the first device, medical device, which could actually do that from your head to toe. But you see that there are no blocks for advancements in visualization. We are quite basic because all the visualization which we do is basically look for like bright red spots and try to figure out if the patient has cancer or not, which is fair enough because that's the reason why people buy PET. But now, if you think about it, we are moving on to a different domain, which is like network imaging, where how different organs interact with each other, and people need, and the doctors and also researchers like us need to get adjusted to the fact that we need to look for different visualization strategies. So as of now, we extensively use kitwest product like ITK, VTK, and we use their medical image visualization throughout. And then we kind of take these results, pipeline it to Plotly, and program it so that we can see some fancy plots and make some interactions with, make some inferences from these plots. And finally, we want to really interact with these plots, and we want to probe organ axis. For example, there are very famous organ axes like hypothalamus, pituitary, and adrenal gland, where doctors want to really see what's going on. So these are the things that we are doing, but what we are actually lacking is a unified approach. As of now, I've been just doing pipelining stuff, but it's very inefficient because if I want other people to use it, it's not going to be that smooth. So our vision, or basically our wish, is to establish these missing links, create a framework which does this by default automatically. Because what I have seen so far is like there are many tools like SPM and FSL. They do a lot of heavy lifting for you, and your job as a scientist is to actually make sense of the data. It actually spoon feeds you all the exploratory data analysis and also this interactive visualization. The only thing missing is like to bring on your own hypothesis and see how it matches to the data that you have found out. So currently we are building this framework called this enhanced framework, which is exploring the human connectome. And we want this to be a web framework because it's easier. We don't need any setup steps. And for example, we already saw that ParaView from kitwest has a web visualization for medical images. And then I found this like really amazing software called Lux, where you feed in an Excel sheet, it comes up with a lot of correlations. It comes up with a set of plots where you can just navigate through it and come up with your own interaction. The final thing that we are like really struggling is this D3JS, because I can actually pick up programming languages quick, but this software is such a nightmare. It's so verbose, and I was not able to build the things that I really wanted to build. So, for example, I would like this framework to have a selection for axis. I want to toggle between male and female cohorts. I also wanted to compare against population. So, like, I want this chord plot to dynamically change for a particular interaction. So this is where we are, and currently we are really looking for collaborators who know what they are doing, because they're used to this kind of advanced visualization. Our main expertise is pretty much on the first and the second one, and not really on the last one. So this is our vision, in the sense that we upload a data set along with their delineations. The software should be able to perform like basic exploratory analysis like scatter plots, distribution, and other things, which is already being performed by this framework called Lux. And finally, this interactive plot where we can see this network connection based on different criteria. So as a take-home messages, I just wanted to say that TotalBody PET is probably the first medical imaging device where you can visualize inter-organ communication clinically, because it has the potential to do so. And we have been developing backend tools for making like really cool data analysis, like automated segmentation, motion correction, and a lot of other modeling things that we are implementing right now. And we have to be pretty honest that our visualization techniques are pretty basic, but even then, for our community, this is not common to have such kind of visualization techniques. And our goal is to build a complex framework that we told you before, so that we do a lot of heavy lifting for the researchers, and the researchers just need to validate their hypothesis. So as a take-home message, I really would like to say that it would be nice to have some visualization experts as collaborators, because you guys know more than us, because we are pretty much imaging experts and engineers. So I would like to finish off this presentation by saying that together we scan, and this is our lab, and that's our usual Monday meetings. We also have a position which I would like to share. We have a new PhD position for an innovative project where we are again talking about visualizing inter-organ communication. So kindly reach out to me or Thomas in case you're interested. Thank you.
Okay, thank you very much, Lalith and Thomas. I think this was really interesting talk, and I'm not the single one, because there is quite some questions in the Discord. So for example, Niels is asking what type of detectors are used in an Explorer system? I'm sorry, now the question popped up. So again, what types of detectors are used in Explorer systems, or is the effective large field of view from the combined usage of smaller field of view acquisitions? And he's also wondering how expensive that might be.
So I can talk with the expense part. Maybe Thomas covers the technical. The expense is 10 million euros. It's quite expensive. It's like fusing a lot of typical PET systems together, but at the end you end up with this big one. So basically the cost increases with the number of detector elements that you use. The other question I kind of...
Yeah, you just pick a question with the money, right?
Yeah.
The detector itself hasn't changed. It's a standard detector that you find in standard PET-CT as well, but it's a 2-meter system. So all the bells and whistles that Lalith mentioned come from the full coverage. It's like really a total coverage, and like a tube in which you insert the human, and you increase your detection efficiency by a factor of 40, simply by the sheer size of it.
I think this nicely transitions into the next question, where Sophia is asking, how do you actually obtain data from healthy volunteers regarding the ethical approval? So how high is the radiation dose in the type of PET-CT scans?
Well, first of all, you can always acquire data from healthy volunteers. These studies that Lalith presented are covered by ethics approval in the US with the UC Davis people. But we are actually launching a study that is governed by ethics approval from the Met Uni Wien as of June. So if you want to experience yourself, not in a 2-meter system, but in a latest generation Siemens system, you can come and join us. The dose depends on the injected activity. For FDG, we are laying eyes in our protocol on about 3 to 5 millisievert effective dose. And the background radiation for someone living in the Austrian Alps is about 3.5 millisievert a year.
Thank you. I hope this answers the questions. The next one is from Nicolas, and he's asking how large should be the training set in order to define a normative network that captures variability in the population, and how do you deal with uncertainty in these cases?
This is actually an excellent question. So for example, for the training data set, we have achieved like 90% accuracy by using 10 data sets, because a lung between you and me is not going to be that different. It's kind of, the variability is very low, and the data augmentation can actually handle this. But for the second question, which is a very tricky question of establishing this normative database, we actually don't know. We don't know how variable it will be. So we hope that certain organs are not that variable like your brain. I can already tell you for your brain, it's quite variable. It's 25% just if I scan you today and if I scan you like 10 days later. It completely depends on your mood. If you're angry now, you'll have a particular metabolic state. And if you're happy on the other day, your metabolic state will be completely different. And that can vary by 25%. But I hope your liver, lungs, kidneys don't change that much. So that's our hypothesis.
Let's hope for the best. So I think one more question here is from Alexander, and he's asking regarding the talks we have heard earlier today about the denoising. So as far as he understands, scattering causes a lot of noises in PET. Are some of the recent neural network-based denoising techniques already used in PET imaging, or is the denoising mostly already included in the image reconstruction instead of applying it afterwards?
So for this one, the Explorer, the noise is incredibly low, because the thing is I think it doesn't have a lot of noise. But if you look for a default one, like a normal size PET, there are noise issues. And we also recently published a paper using deep learning, using conditional GANs, where you could denoise the whole setup. But this is not typically encouraged in clinics, because what we found out was once you apply denoising using AI, the values are not really that accurate. The typical denoising methods based on classical approaches work much better, but nothing is really put forward by the community right now. I just know that GE has one, but it's not quite common, this denoising approach.
So there is one more from Anya. How do you compute the dependencies of the different organs?
So right now it's temporal correlations. We look for we exactly mimicked what fMRI people did, because we didn't know how to analyze the data. PET people, we have not done this before. So we calculated the temporal correlations, and we actually said, okay, if two curves look similar, they should probably have some kind of relation to it. We don't know if it's actually true, but we are working on paradigms where we can actually prove that, okay, if a particular curve looks similar and after like 5 seconds, you see another organ expressing the same curve, we think that they might be connected on the basis of Granger causality. So that's what we are aiming at. So basically the answer is temporal correlations.
Thank you very much. And the last one is from Ernesto. So could you theoretically label cells in the context of therapies and monitor their movement?
No,
yes.
Our resolution is right now.
It's possible. There are.
No,
Go ahead, Lalith.
No, I mean, it depends on the tracer. If you, I don't know, can you hear me?
Yes, we can.
Yes.
Yeah. So there are two things for this. FDG is just one thing that we are doing right now. For if you want to track a particular cell, you just need to radio label them with a code. I think that's what Thomas wanted to say as well. But what kind of cells are you guys interested in? That's the question.
So Ernesto, can you provide more information?
Probably stem cells.
So immune cells.
Okay.
Yeah, we can do it. Yeah.
Okay. So I think that was the last question I can see here. If there are some more questions popping up, please write them later on within the Discord channel. The Discord is going to be open for the whole week. So if we spot some questions, either the speakers or we will grab them and try to answer them as best as we can.